This might be the craziest graph in biology. For billions of years, life stayed small, like bacteria. And then 500 million years ago, we had the Cambrian explosion, which created all of this big life, plants and fungi and animals. And even after 5 mass extinctions, we got mammals and then us Homo sapiens. So today we're going to tell the story of this chart. And this chart has 4 big puzzles in it. Okay, the second question is, how do genes create body plans? In other words, how do we get these endless forms most beautiful? The fungi, the plants, the insects, the octopi, the humans. And the answer is that we have these genetic controllers that efficiently turn an embryo into a responsive, modular 4D map of tissues. There's a lot in there, but all of it's important. The cool part is that we have these genetic controllers, which they're called Hox genes, or they're called transcription factors, and they take up about 20% of our genome. And what they do is they tell us when to turn on and off other things. Because remember that my body is 30 trillion cells, but they all have the same genes in them. And so the only way to know what each of them do is by having these genetic controllers. And those genetic controllers, they efficiently turn an embryo. And what I mean by this is we have this little embryo. All animals come from a little embryo, a few little cells that then turn into something much bigger. And what I mean by efficient is that there's a whole set of things that make it so that death is kind of minimized or happens early in an embryo's life, and that the kind of variation that you get off with little fingers or different colored eyes and things like that, that the mutations are able to create lots of variation in the body plan itself without killing me. And what does that body plan look like? It looks like a responsive modular 4D map of tissues. So what that means is that this 4D map is saying, hey, when you're building a body, that body is, you want to know where to build the limbs. You know, if I'm a snake, I don't want to build any limbs. And then you also want to know when to build them, you know? So it's like when I'm a kid, I'm not supposed to build some things until I hit puberty, and then I'm supposed to grow a lot taller and things like that. And so this system of genetic control, also sometimes called evolutionary developmental biology or Evo-Devo, or it's sometimes called embryology, they create all the different body plans of multicellular life. So let's dive into how this works now. Okay, so let's first situate where we are in time. Remember that we just got to the Cambrian. That's amazing. And now we're in our final eon, this Phanerozoic eon. There's the Paleozoic with some of these old school creatures. There's the Mesozoic with all the reptiles. And then there's a Cenozoic where we are today. We're in this tiny little thing at the very end here. And this overall is about 500 million years. And during this time, we get all these amazing creatures. And I love what Darwin calls this. He says, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. I love that. Thinking about them as endless forms most beautiful. That's what all these body plans are. They're just like, what? This is crazy. We created all this seeming design out there? It's so cool. Evolution is cleverer than you are, and evolution produces such cool things. And so you can kind of see that shift with this graph here of biomass composition. This is specifically marine biomass. And you can see at the beginning, obviously 4 billion years ago, when life started, it was mostly methanogens and subsea microbes. And so these are some of these creatures in those deep sea vents or whatever. And then over time, we start to get, you know, those sulfur bacteria that we were just talking about, the sulfur-reducing bacteria, and also the sulfur photosynthesizers, the anoxygenic photosynthesis. So finally, around 2.5 or 2 billion years ago, we get the Great Oxygen Event. We start to get some of these cyanobacteria. And then finally here, we get these algae that we were just talking about, where you get a bunch of these algae, and then those enable all of the animals that we know here in blue. Here's another way to view that graph, but as a total amount of biomass, where you can see the biomass just increase so much, where you can see we've added all of this algae and cyanobacteria, and then also all the animals on top of that. So how do these endless forms most beautiful work? How does genetic regulation happen? Well, the best way to start to understand this is to understand the main 9 animal phyla. So we definitely have fungi and we definitely have plants, but thinking about animals and thinking about the phyla that exist will help us understand how we make all these different body plans. And the great part is that the phyla are broken into these body plans. And so here are these 9 main phyla, and it starts some of these old school ones down here. We got the sponges that we started with, then we get the jellyfish, the Cnidarians, and then after that we get, which are these creatures that have bilateral symmetry, like us. And so you have roundworms and flatworms, you have mollusks, you have starfish, these echinoderms, and then finally after that you get these segmented animals that have these little segments, like little segmented worms, or arthropods with their little segments, the insects with the wings and the claws and stuff. And then finally, vertebrates like us, these chordates. And so what we're going to do now is kind of go through these, go from the sponges to the Cnidarians to these Bilaterians, and then finally to these segmented animals. And you can kind of see that there's at the beginning, we have a cellular level of organization where you just have a bunch of cells kind of hanging out being a sponge. Then those cells form into tissues like a jellyfish that has like an outer layer and an inner layer. And then finally, we start to arrange those tissues into organs. And those organs are arranged in a creature with bilateral symmetry. And then finally you get segmentation, where you have these different segments of the body plan. So yeah, let's start with sponges. Sponges, they're amazing. Periphery. And the way that sponges work is they essentially have these little flagella here that are flailing, going blah, 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 blah. And that is kind of bringing water through them. And as that water goes through them, they eat it here, and then the water goes out the top. And so you can see why sponges are at the start of life. And so there's no real inside or outside here. It's just a bunch of cells all working together. And you can see that here with a set of images that shows the transition between an ancestral thing called a choanoflagellate and then a modern sponge. And so you can see these choanoflagellates, they're essentially just a combination. Each of these is a single cell. And so each of these is a single creature, a single-celled creature with its flagella going behind it. And multicellularity evolved tons and tons of times because it just made sense. You had all these creatures that kind of form together. They form in this way where then they start to do a little bit of division of labor, where they say, oh, we're kind of forming like this. What if the inside, what if you guys just hang out while the outside does kind of the movement? And then you can start to see, oh, maybe these sponges start to move in this way. And oh, here's an advanced sponge. And then this one's like, oh, what if the inside things are doing this? And then you get, oh, what if you have kind of a gastrulated area, kind of an inside? And then boom, before long, you see something that looks roughly like a sponge. And so here's a image that shows a phylogenetic tree of some of these really old species, like sponges. And you can see here's how we start to get sponges up here, and things that are connected to them are these choanoflagellates, where you just have an aggregate of cells that are kind of doing the same thing. But the other way to get at it is to say, oh, maybe you have this thing that's kind of like an embryo. So here's an image that shows the kind of two ways to get to a sponge, either by combining lots of single-celled creatures into a bigger one, or you can take a single-celled creature and have that creature make copies of itself, but instead of having them go out into the world, they kind of get stuck together. So that's kind of what an embryo is. You're making lots of little cells, where you're starting from one cell, a zygote, and then you're making it into 2 cells, and 4 cells, then 8, then 16, then 32, and before long you have 30 trillion cells. And so this was happening around the time of sponges a really long time ago, where these little creatures are creating multiple cells with nuclei that are still part of the same structure. It's another way to get to multicellularity. Here's another image that shows that, where you can see these little things are breaking apart from each other. You're getting all these new nuclei, but they're staying a part of the overall whole. And so sponges do this when they reproduce. They have embryos. They do a bunch of cleavage to create, a 16-cell thing, creating a blastula. That thing kind of swims around, it gets inverted, it flies out into the world, it then kind of reverts itself again, and then boom, before long, you have it kind of sets its little larva down onto the floor of the sea, and you get a new sponge. Here's another image that shows this across a bunch of different kinds of sponges, where they have initial cleavage, which creates a blastula. You get the morphogenesis into these different forms. You have your little larva that swims around and eventually creates a new sponge. Okay, so those are sponges. They're just essentially a bunch of choanoflagellates hanging out together doing division of labor, but they do have an embryo and they all are multicellular. Now that we can look at the second phyla, Cnidarians, or the jellyfish, and they have 2 big differences. One is that they have radial symmetry, and the other is that they organize their cells into tissues. So here's just a quick image showing that, sponges, periphery, no symmetry. But then Cnidarians do have radial symmetry. Their embryos are organized to be radially symmetrical. And I just love showing these pictures of some of these phyla. And here's the 4 main kinds of Cnidarians. You have jellyfish, which are crazy, flopping around. And then you have these little hydras. You have the anemones that kind of zap people. That's the key thing with Cnidarians, is that they have these little ctenophores that kind of zap people. And then finally corals. I was surprised to learn that corals are also Cnidarians. They have each of the little polyps is radially symmetrical, and then they kind of organize into colonies. And then here's what we mean by tissue level organization. With something like a Cnidarian, they have an outer layer called an ectoderm, and then an endo layer called an endoderm. And you can see here that if I'm a jellyfish, I have my little mouth here, and that mouth is both my mouth and my anus. It's an opening to then put stuff into my body, digest it, and then push it out. But then I have the rest of my body, my skin, my ectoderm, which protects me. And so something like a jellyfish, these Medusa, these Cnidarians, and they're a bag, they're just 2 layers, so they're called a diploblast. They only have 2 layers of tissue, while someone like us, we're a triploblast, we have 3 layers. Okay, then the next part of the body plan that evolved was Bilaterians. And these are creatures that are bilaterally symmetric, and they have organs. So you can see here are these examples of bilateral symmetry. You have sea turtles and butterflies and fruit flies and lobsters and humans. They are all bilaterally symmetrical. It's amazing. Here is the first creature, a little flatworm, that was bilaterally symmetrical. Cute little guy. And the other thing these creatures have are organs, and then also this mesoderm, which is this kind of middle layer. So you can see, here's a worm that has a tube within a tube body plan. And so the tube within a tube, we're all tubes. And so I have my mouth here, right? Here's the little earthworm mouth. And then there's that tube goes through, and then boom, there's your anus in the back. I got my butt in the back. And so the body around me is kind of the physical tube, and then inside of me is my gut or my other tube. And you can see here that there's the outside, the ectoderm with the body wall, your skin, things like that. There's your inside, the kind of your gut derived from your endoderm. And then there's this weird middle area where you have muscles and organs derived from the mesoderm. So this is a middle area with the thing called a coelom. And that coelom is essentially a place, when you imagine where your heart is and where your lungs are and your stomach, that stuff, it's not in your skin, but it's also not in your gut. It's kind of in this weird middle area. And so that was the key thing with these new creatures. They had a place inside of us that didn't have a job to do. It didn't have to be a gut, and it didn't have to be skin. You could say, hey, you're just gonna do you, and whatever the inside of the body needs, let it go be an organ. Here's an image that shows that same tube within a tube body plan that exists for insects and vertebrates too. And then here's an image that shows those different derms and what they create within humans, where the ectoderm creates the skin, it actually creates our brain and stuff too. The mesoderm, the thing that creates all of our skeleton and muscle, and then the endoderm creates our gut, but then also everything that goes in the mouth, like our lungs and things like that. Okay, and then finally, we get these segmented animals up here. So these are earthworms and arthropods like insects, and then also chordates, aka vertebrates. And so what these things do is they start to segment the body into different places where you can put limbs. And they do that with what are called these Hox genes, where you can see that whether you're a fly or whether you're a little notochord creature or whether you're human, we all have the same little segments in our bodies that then determine, oh, what should be your head? What should be your thorax? What should be your abdomen? What should be your limbs? You know, for us, we have a similar thing where we say, hey, this part, you should grow limbs here, and you should grow legs here, and you should make a little ear or whatever. So this is what happened at the phenotype level, where these are the different body plans that get made. And I just love this image because it shows kind of how simple all the different body plans are, and also how they're a little bit different from each other. But now let's look at what happens genetically in our embryos that enables these different body plans. And so as a reminder, this always happens through an embryo. Just like we do with human bodies, where I see my friends and they have these ultrasounds and you can see the little creature starting to form and it gets bigger and bigger and bigger. And then boom, 9 months later, you have a baby. That happens for all of these different animals as well, where you start with one cell and then the embryo gets bigger and bigger until it turns into the actual creature. So the way this works is that as you go from the small stage up to the full body plan, all of the little cells are talking to each other. At the beginning, they're saying duplicate, duplicate, duplicate. And then they're starting to say, okay, you guys over here do this. You guys are going to be the skin over there. Oh, you're going to be the front. You're going to be the back. And then before long, they say, boop, 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 and now I'm a sponge. And remember that, and it doesn't do this through a computer or whatever. It does this through chemical signaling, because that's all that biology has. And so it does it through what's called these morphogen gradients. A morphogen are these little cellular signaling molecules that one cell will push out to a different cell, and in the other cells it kind of trickles out over time, and then maybe the cells over here are like, okay, I'm going to be the inside, and the cells over here are like, oh, okay, I'm going to be the outside. And so the way I like to think about this is that the cellular signaling, those morphogens, have to give the 3D body plan. And so some of them do bottom from top, where you can see something like BMP is one of these signaling molecules, and cordon is another one. And what that's saying is, hey, you know, the dorsal versus the ventral. If you're like a shark or whatever, your dorsal fin, there's things to say, this is going to be the top, and then this is going to be the bottom. This is true across all species, where you can see those are the ones that are used with chordates and frogs, while this DPP versus SOG, those are the ones that determine the body plan. But you also want to determine head from tail. And so this thing called WNT, that's the thing that shows, you can see the green is where the WNT is. That's being like, oh, here's your tail, here's your tail, here's your posterior, here's your back. And then the A is the anterior. That's if you have a WNT inhibitor, you're saying, hey, little mouse, at your front, none of that, but at the back, okay, that's cool. So you have across all these different species, they're all using WNT to determine their head from their tail. The other part that you need to do is the Z-axis, where you need to say, what's the inside of me? What's my endoderm? What's my mesoderm? What's my ectoderm? What's my skin? And this is often done through this thing called nodal, where the nodal is saying, okay, here's what's going to be kind of mesodermy, and then it starts to produce these different derms in the early embryo. And so what you're left with is an image like this, where you see you're trying to make a little tadpole here. And the way that you do that is you start with an embryo, and you have a certain thing that's determining endoderm versus mesoderm versus ectoderm. You have your ventral versus dorsal with something like BMP. You have your posterior versus anterior with this WNT thing. And so these morphogens are signaling to each other, eventually turning into this tadpole that has a 3-dimensional body plan. And then finally, if you want to, you can take that 3D body plan and break it into segments and then put limbs and things on those segments with Hox genes. These morphogen signals also create the organs, where you start with some of your little simple signaling to create the derms and things, and then you have your midgut that then can turn into, with some different signaling, it can turn into the liver, it can turn into your lungs, it can turn into your pancreas. Or, here's an image of the development of the heart. It's essentially just a bunch of tubes where where the tubes are talking to each other, and they eventually create your atrium and your left ventricle. And then before long, you have your 4-chambered heart. These morphogen gradients can also create other amazing things. So this is, our hands. Here's my hand. Boop. This is created where you start dropping a little bit of a signal on one side, Sonic Hedgehog, it's the name of this thing for some reason, and that over time, it tells you what should be your fingers, and then your thumb gets created because it's like, oh man, there's not much SHH here, guess I'm a thumb. And then similar with these amazing patterns of skin, and these are created through something that's called a Turing pattern, which Alan Turing, obviously, total polymath. And he came up with this idea in like 1952, way before we even knew about genes or the DNA helix or anything. And he said, yeah, if you have these chemicals interacting with each other in a certain way, you can get these patterns of on-off behavior that we see with scales and with these amazing fish and, kind of leopard patterns. And all of this is part of what's called an algorithmic phase transition, where you can see what this graph shows is how long the genes of different animals are, and then how long their average protein is. And you can see that for a while, with these little creatures, with bacteria and archaea and protists and fungi, they were all trying, they got bigger and bigger gene lengths, but then those genes mostly just made proteins. If you have a gene of 1500, each one codes for, it's 3 genes to code for a given protein, and so 1500 divided by 500 is 3. And the reason for this is because you get these big proteins, but it doesn't help that much to get massive, massive proteins. At this point in time, what life started to do is say, if I make bigger proteins, it doesn't matter that much. What I need to start to do is to find different ways to get those proteins to interact with each other. And that's when we get all of us over here, the invertebrates and the vertebrates, where all of our mean protein length is the same. It's kind of leveled out at 500, but our gene length is really long. And what do those genes do? Those genes just talk to each other to build a body plan. So this was a crazy thing when we initially sequenced the human genome, and we thought it was going to have a really, really long genome, but it was only 20,000 genes, which is like the same as these other creatures, as these dumb creatures. And we're like, what's going on here? It's like, well, what's going on is that they're all just using 1,000 or 10,000 genes to kind of build a bunch of tissues and organs in a body plan. So we were kind of sad when we were like, oh my God, we shared 90% of our genes with chimps, and we shared 62% of our genes with chickens. And and 28 with a fly and 17 with a banana. It's because a lot of these genes get frozen out and the only genes that are left are some of these little ones that are specific to the creature. I love thinking about this as, so what is the morphology of a creature? What does its body plan look like? And you can see here's an example of creating different conch shells, different shells, and some of these look like nautilus, and some of these look like other little mollusks and things. And this is all just computer generated, but this is essentially what evolution is exploring. It's exploring which of these different forms most beautiful actually work in reality and in a given niche. So I want to conclude this portion with this amazing screen grab from this great talk that this guy Mark Krishner gave, who came up with the idea of evolvability. And I haven't been able to find this image online, but there's a talk that he gave in the early 2000s. And what he shows here, he's like, hey, here's what really happened over these 4 billion years. Innovation came in waves followed by regulatory change. So it's not like we had random innovation over time. No, instead, we had these waves of innovation. And so at the beginning, we got the origin of life. We got metabolism. We got DNA. And all creatures use that. This got frozen out. If you change how your metabolism or DNA works, that embryo is going to get killed and never going to turn into a full adult. Then we got these eukaryotic cells, and those again got frozen out. We got the origin of multicellularity, what we have today. That got frozen out. Then we got the origin of these complex body plans. We are trying out all these different body plans. And then finally we got these different origin of limbs, where it's like, oh, you can be an insect with these limbs or a vertebrate with these limbs. And I love what he says about this transition here, where he's like, yeah, if we essentially got those 10 phyla at the beginning, and they haven't changed much. And so if you were a creature later on trying to make a new body plan, it's like, no, that already happened. We've already made the Industrial Revolution and the internet and stuff like that. It's like, you're supposed to work on AI now. But so transition had moved on to segmented creatures, exploring Hox genes, and exploring the different ways you could organize limbs, either with exoskeletons or with a vertebrate kind of backbone. So how do genes create body plans? These genetic controllers efficiently turn an embryo into a responsive, modular 4D map of tissues. Thank you, as always, for watching. And thank you to Roots donors for supporting this channel. If you want to understand more about the past and how we got here, check out the previous videos here. And if you want to understand the future and where we're going, feel free to subscribe here. Hope to see you then. Bye.